And we open with a talk from Olivier, who's come here from the center of unknown. so Olivier is actually in charge of basically creating the sort of like informatics infrastructure for the IBL, International Brain Laboratory, which is basically an initiative to have a set of experiments done almost uniformly across a set of labs. And with neuropixels and huge amount of data, of course, is generated by IBL, which makes it very challenging to like maintain this data, curate it, and also do like a knowledge discovery in this sort of data set. So, Olivier.
Thank you. Yeah. So, as software engineers at IBL, our mission is to maintain the dataset and to distribute them. And we started doing this 6 years ago. And of course, since then, there's been kind of an AI revolution. And many people are asking us now to use the data in order to train those models that we were mentioning yesterday. So a big question is, what has changed? How do you cater to data-hungry modeling attempts? And I can tell you right now, it's not very different from what we were doing before in terms of good practices of how to distribute the data. So throughout this talk, I'm going to describe a bit what are the good practices, the good principles in order to have open and maintain an open data sharing, and what are the very particular things that can come for those modeling attempts, which is mainly about scalability. So I'm going to focus on the 3 really big requirements, which is quality, reproducibility and traceability, and the last one is how to scale the computation. Before I get into the details, I think it's nice to give a bit of context. So I'm going to present the IBL task because this is important to know that we have a single task and all of the data that we have is kind of centered around this task. And the task is a vision decision-making task that is very well known from Carandini lab. There is a stimulus that shows up on one side of the screen. The mouse is head fixed, and the mouse has to move the wheel on a cue, and to move the wheel to put the stimulus in the middle. If it manages to do so, it gets a reward. Otherwise, it doesn't get a reward. On the right, you can see that the aggregated data of electrophysiology recordings for the brain-wide map. And you can see that at you can see during the trial the activity that is changing according to the behavior. And here what I want to expose is the fact that this is a single task, but this is very large region recordings and very varied region throughout the brain. The next thing to understand is to see a bit the context of those efforts is we can look at this in the project side. So in terms of research, we can look at all of the projects that are around this task. So the main one is the brain-wide map. It's the flagship project, the decision-making task. The second one is the electrophysiology atlas, where we try to look a bit at what are the electrophysiological features in the context of the brain anatomy and where you are in the brain. Another effort going on right now is a mesoscope acquisition, where we try to image a large part of the dorsal cortex within the same task. Another effort is concern multi-area simultaneous recordings. So in this case, we identified 16 areas according to the connectome. And the idea is to record those simultaneously to try and compute the dimensionality of the communication subspaces. We have efforts going on into the behavior modeling. So here it's HMM kind of models that will try to compute some behavioral states that we can then relate to neural activity. We have some neuromodulator efforts where we try to record several neuromodulators in the same context of the task. And at last, the latest example projects are the large neural models trying to train some kind of foundation model for the IBL data. So all of this we can see as well in the context of the modalities that we record. And this is a many-to-many relationship between those projects and the modalities. And notably, the 3 first modalities are common to all of the projects. But then we will have some specific modalities depending on the projects, such as electrophysiology, photometry, and mesoscope. So all of this, here I'm going to present those things in the view of the data requirements that I have just cited before. So quality, reproducibility, and scalability. And each time I'm going to illustrate my point a bit with scientific projects and to motivate a bit the purpose of those requirements. So one last thing I need to do in order to set the context is to understand what is the architecture of the IBL. The IBL is a distributed collaboration, which means that the data is acquired in separate parts in separate labs, and we need to integrate this data somewhere. And so it's 11 experimental labs, and the data size is around 450 terabytes. But this is a lot of sessions, 40,000 experimental sessions. So right away, 6 years ago, we had to go with a way to consolidate the metadata in a single place. And for this, we have a colony management relational database that is served as a web app, and where each of the experiments that go on at IBL are actually registered in this web application with all of the metadata. This is really similar to what Saskia presented a bit before, and I think we scored quite well on all of the requirements that Saskia presented. All of this is in this relational database. The data files themselves are not in this relational database. They are in San Diego, in the supercomputer, and we have an Amazon S3 bucket as well that mirror this master copy at San Diego in order to serve the data a bit in a more easily fashion. Also, worth noting is that when we export a project, we usually export an NWB version of the project and to Dandy. So I think I've set up the decor right now, and we can get to the meat. So we'll start with the quality control. And in order to motivate a bit the quality control, at the very beginning of the project, we decided to say, you know what, we are going to try to reproduce the same insertion, same experiment in 11 labs and see how reproducible this is. And so we gave micromanipulator coordinates that you can see in the left, and we had 11 labs reproduce this experiment and this insertion. And as you can see in the reconstruction of the histology, we are quite far away. Here, this is 1 millimeter. So it's really hard to target the insertions. The second thing that we saw is that electrophysiological features are reproducible across lab. So that's here you can see on the top, this is the PSD of the LFP across all of the labs, and at the bottom we are looking at the firing rate. And we tried to train a machine learning model to try and decode the lab from this data, and we found out that we couldn't decode the lab identity above chance. But we had to enforce quality control metrics in order for this to hold. And so we had worked quite hard on all of those quality metrics depending on each of the modalities. And here there's the plot of the original insertions that came in the project and the ones that came out and the reasons why they were removed. So we have some automated metrics. Some are due to the behavior, some are due to the yield of after spike sorting, some are due to histology or hardware failure. So let's dig a bit more into one modality to see a bit how that works. And this is the electrophysiology modality. And here, this is a table of the metrics that were used in this specific case. What I want to draw your attention on is you have raw data metrics versus spike sorted metrics. In spike sorting metrics, so we have the cluster-based metrics here. They are computed. But it's a very different meaning. If the spike sorting failed, it doesn't necessarily mean that the raw data is bad. We have automatic metrics versus visually assessed metrics. Some of the metrics can be automatically computed and thresholded, and the other one have to be assessed by an operator. At last, we have 3 metrics about the clusters here as per spike sorting, but we compute many more. If a metric is enforced, it becomes a criterion, but it doesn't mean that we don't compute other metrics. We can follow this up now with a video QC, and it's a bit the same story. Here we have raw data metrics automated. We compute the frame rate, sync, and the contrast and the positioning of the camera. And we compute pose estimation metrics. How many nans do you have, for example? What is the confidence? And is it in sync with task events? And this is the display that we have in this relational database, in this web app for each experiment when we have a DLC running. And we can check the quality of a given session, looking at the pose estimation result overlaid on the camera results. And especially looking at the extracted pose features as a function of task events. So if we try to sum up, we have several modalities. We have the same for mesoscope and other modalities. So we have a lot of metrics that goes into this relational database. So the first thing is to monitor them. It means to put them into the database. But the second step after monitoring is really about reporting. So, and reporting, we need to aggregate a bit those metrics. So we have dataset level metrics. And then those dataset level can be aggregated and to be at the recording level metrics. So here, AP band, LF band. Then we have the session level metrics, the one that concern the behavior, for example, or these kind of things. And you can see this a bit as a funnel at the end to have a session aggregate metric that you can query. And you can query at all of those levels. So it's really what Saskia said in the fact that if you're interested in electrophysiology, maybe it doesn't really matter if the behavior is failing. So here you have the possibility to query the database for all of those levels of metrics. So what is the lesson learned about those metrics? That's exactly what Saskia said. One man's trash is another man's treasure. So failure of behavior does not mean that the EFIS data is non-usable. Even in the EFIS data, AP band data sometimes is good while the LFP is failing the metrics. What do you do with seizures, with epilepsy, for example? So it could be interesting for someone, but you really don't want to decode this in the context of the task. Another thing is that failed process data, if you have a bad spike sorting QC, it doesn't mean that the raw data is wrong. In the end, external users, many users want a single number, but it's really hard to aggregate. And here what we do is we try to sign off by modality. We can say, okay, for this session the video is good, for this session the EFIS is good, for this session the behavior is good, and then the user can make their own decision about which data they want to use. So this is Easter, so I made a little Easter egg on the previous slides to expose a bit to an anecdote about the data QC. So let's go back a bit. And here, if you look at this DLC QC, you can see that it is failing. It is failing because the pole position has not been computed properly. And if you look at the corresponding data, what is happening is that we have this mouse on the left camera, it's holding one paw back like this. And it's doing this throughout the whole session. Most of the mice use two hands to move the wheel. This one uses only one. It's like we call them a truck driver mice. And there is an occlusion that happens throughout the session, which means that we cannot reconstruct the pose of this paw. This makes the QC to fail, but of course, this is not a session we want to throw away. This is inherent to the data itself. So automation is nice, but you will have some fallback, some problems with automation. And I think if I could go back in time, I would really abide by this. I would work more on visualization than automation. I would put the visualization first, automation second, and to have the QC as early as possible, and to train the experimenters to look at those QC, so that you build expertise in your collaboration. The last thing, and it's kind of a detail, but it's important when applicable, save the label processing outputs, confidence. You can save them with your data, directly with your data. For example, the cluster quality metric should be a data set. DLC outputs the confidence metrics for each of the pose estimation. So the second point, that's for quality assurance. The second point I wanted to mention is the reproducibility and management of change. And this is a bit about traceability of versions and revisions, and why do we have those revisions to start with. And this is really, if there is something I would like you to get away from this talk, is that those data sets are alive. The spike sorting is not really a complete method, and every 5 years or so, we may want to reprocess the full data in order to get a better output. And this is what happened a bit last year. After 5 years of incremental changes on the spike sorting pipeline, this is the list of incremental changes. So there's small changes, like if you start running spike sorting on 1,000 of NP1 probes, you will start having problems that people have not seen before, and you will have to tweak a little bit the normalization of the widening matrix. You will have to change a bit, in some cases, the motion estimate for the drift correction. You will find some weird bugs. You will adjust some detection thresholds. You will clean the waveforms. After 5 years, many a mickle makes a muckle, and it makes quite an impact if you rerun your pipeline at this time. And here, this is an example of the yield we get with the new spike sorting version per brain region. All of the yields increase across the brain. And it's the same for a simple decoding score on the right, so the p-values decrease while the decoding score increases throughout the brain with this new spike sorting. And then you get into the case where you need to use data revision. You need to revision your data. You need to make sure that your users are aware of which data they are loading, or if they have written a paper, that people can reproduce this with the actual data that they've used for the paper. And this also is part of the metadata database. And this is saved. Each dataset has a unique ID and is saved in the database. The next part is a bit more related to how to scale. So everything is harder when you start scaling up. And the first thing is really similar to what Saskia said, is that you need to have discoverable datasets. So for us, it's quite easy because it's a relational database, so it's kind of straightforward to make queries. But here is an example of if you want a query of the 550 sessions or so, 700 recordings that constitute the brain-wide map. You can go to the website and you can search for a region. On the first day, we had some aficionados of the medial septum. So here is what you would do. You would enter medial septum region and you will see the probe insertions that actually went into this region if you want to have a look at those specific data sets. Of course, there is an API as well that you can do with code in order to research exactly and to get the dataset IDs that correspond to this. The next step that is important when you try to scale up is that you need to make sure your datasets are randomly accessible. And this I'm going to go back in time 6 years, where we ended up with the electrophysiology, and we had the problem of compressing the electrophysiology, and nothing existed at this time. What happens is that you can less compress NP1 and NP2 by a factor of 3 and 2, respectively. So that's great. But one thing that is really important to understand about compression is that if you compress the whole thing and you put this into a drive, of course, if you want to access 1 second of data, or if you want to slice the data, you have to download the whole thing and compress before you can access it. So we really insisted that in our requirements back then to have chunked compression. What is great is that it became a standard in the field now with ZAR. ZAR at the time did not exist. It's a standard in spike interface, and it's used in other electrophysiology pipelines. But this is really important if you want to have your data easily accessible, is to make sure that it's sliceable. The last part of the actual scalability is how do you prepare your data for analysis and how you make it easier for neural analysis. And here, I'm going to go through several levels of data preparations for several complexities of analysis, of computations for analysis. So the first level, let me go back to the task. I'm going to talk a bit about the brain-wide map data. So the stimulus, you can operate on the contrast of the stimulus to have easy and hard trials. You start with 50-50 probabilities for each side, but you can also have then add some blocks where you have a probability that the stimulus is much more likely to appear on one side than on another side. And then you can look at the modulation of the psychometric curve as a function of the block. Yesterday we spoke a bit about Bayesian brain modeling. This is a bit what this is here, and we are trying to decode what we call the prior, but which is actually the block probability from the data. So that's an example. You take one session, you take a session that is in this orbital region. You have the base optimal prior, and you try to decode the prior directly from the neural activity. In terms of computation, this is not very hard. It's embarrassingly parallel. You have one job per session. You run this. It's easy. You can run one session on a laptop. For statistical reasons, we want to bootstrap, so we need to run this thousands of times. So it's a bit expensive computationally, but there is no complexity. So this is level 1. So we have another level would be, for the example, the IFIS atlas. For the IFIS atlas, we want to start extracting features from the raw data, electrophysiology features, and relate them to where they are in the brain. So here is an example of all of the raw data snippets that are taken from many different regions. And as you can see, it's wildly different from region to region. We are looking at 50 microseconds of data for 384 channels here. Olfactory bulb, there is a big party going on. Like in the cerebellum, it's much quieter in the hippocampus. As you can see, it's very different. So the question is, can you extract useful features from that? So here the processing is going to be a bit more complex because we need to, for 1,000 recordings, which is hundreds of terabytes, you need to start doing some kind of pre-processing and extracting the features and put them into a table. There are several pre-processing jobs and several extraction of features. There is a spike localization part that runs on GPU, so we need to handle different environments. So here we start handling job orchestration systems and this kind of map-reduce architecture. So here we output 24 features for 365 channels. It's hundreds of megabytes. There was a reduction of the order of 1 million before where we started, where we ended. Is it useful? Yes. If you look at the features as a function of the brain region, you can see that you have clearly signatures of where you are in the brain depending on the feature. This is the peak to through duration, peak to trough duration. You can then train a decoder, for example, to try to decode the brain region as a function of those electrophysiological features, and this is more or less what you will get. So this is the prediction on the left, this is the real region where you are, and those are the cumulative probabilities. You have to live by your standards. I said earlier that you need to save the actual probabilities and the uncertainty, and this is what we save as an output. This is much more informative about what's going on than the simple most likely prediction. The last thing about this project is that it's multiple modalities, and we're integrating this with genomics and the Allen CCF Atlas and the IFIS features, and it's really interesting the relationship we have between those modalities. The last level is really you have to stage the data. You have to prepare the data, to duplicate the data, transpose the data in order for it to make an analysis that is much more powerful. So here we are going to look at training over 2 million choices. And the idea is that if you do this session per session, it's going to be very long to run your query and extract the data. While if you stage the data in a table, you can run action kernel learning rates aggregation in a matter of 20 seconds, while it's something that will take 10 or 20 hours on the raw data itself on the sessions. So you're going to tell me, great, but you haven't mentioned those large models yet, right? But I think you have all of the ingredients here and what you need to do in order to be to scale to those large models. And if you look at what people are doing and what we are doing at IBL in order to load data into those transformer training batches, this is exactly what they do. They slice the data. They make sure that all of the modalities are sliceable, wherever they come from. And also they make sure that they duplicate the data in a format that they can access very quickly in order to feed batches to their training transformer. So this is an example from temporal data, which is a tool that is preparing the data in order to be run in those models. I'm going to conclude now. If I have some practical recommendation on how to prepare, how to maintain open data repository, is to capture and store metadata in a machine-readable format. It really goes with what Saskia was saying before. But then annotate them with QC, and annotate them with data revision, and provide reports and querying capabilities on those things. Second thing is plan for random data access. Make sure that people will be easily able to access some random part of your data sets, because you never know what people will want to do with it. And the last thing, if you want to do analysis at scale, first plan your analysis once you want to launch it. You will have to consider job orchestration if you want to be efficient, and at last you need to consider as well how to stage the data in order to best fit the analysis to have a quick iteration cycle. With this, I'll thank you, and I'll take any questions if you have.
All right, questions? All right, Yarek. Okay.
In that slide, I missed it. That variability in place, how much could have been attributed to just anatomical difference between samples in months?
Yeah, it's a good question. I would return the question to some people here, because with this MRI thing about how, so the question is how much MRI brains are varying as a function of the Allen template. So I don't know.
Yeah, most of it is we don't know where the brain is within the skull with respect to skull landmarks. No, that's the sort that answers your question. to first.
Brains are also different.
Well, that's kind of the same. We don't know, right, sure, brains are different. That's maybe a different way of putting it. And skulls are different. Yeah. So the relative location of brain regions with respect to skull landmarks varies from across individuals. And there's the other issue that's highlighted. I think I'm not sure if you've mentioned that, but there's also sometimes vasculature that makes you shift from one place to another. Since I have the microphone, I wanted to ask you a question. The brain atlas. I mean, it's great to recover brain regions, but these are big compartments, right? So the question really is, what limits you from, and it's not like you would miss those, right? So you sort of know where you are based on targeting, but how granular, what limits the granularity? Is this data, or is it actually the lack of features?
I think it's more the data right now. The problem is that the alignment we work with to start with are not perfect. So there is a whole cycle of data refinement. The second thing is that not all brain regions are necessarily different from their neighbors in terms of IFIS features. So here we could try and find the optimal parcellation of the Allen regions that would maximize the entropy of the IFIS features, if that makes sense.
Yeah, I mean, as you heard from Lydia earlier, probably hanging your hat on the Allen regions is not what you want. You want this to do this in a data-driven way, right? You want to make the next generation of Allen regions based on this kind of data. So, good. But I like the answer. Thanks.
All right, yeah, actually, would you mind passing the microphone to Jarek?
I could start. So it really struck me. Automate is nice, but visualize come first. And I love automation. And is that just because we don't have good metrics to extract automatically from those videos, maybe, and the AML could help, or is there more to it?
This kind of it's really hard to predict what's going to happen. So this two hands, one hand thing is not something that we would have forecasted, and you need to look at it in order to understand what had been going on.
So it's like with coding. I mean, after you detect it, you need to write a unit test,
yeah,
right?
Exactly.
So it's like, and then implement it, but you need to detect it first. And one more, if you don't mind. For the regions classification, did you look into kind of flipping it sideways? Take two regions which you know they're different and try to claim classifiers to extract the features which are important to tell them apart.
Yeah, and for this we use the genomics. We use the AGA atlas to do this kind of encoding. So that allows us to do some kind of co-kriging of our measurements into the whole atlas, and then we can ship full features volumes.
Did you look which feature is important to tell areas apart generalizably across, let's say, experiments?
Yeah, feature importance, you mean, in the classifier? Yeah. LFP. LFP works very well.
Okay. Jerome?
I'm really curious about how you coordinate the downstream analysis across the lab. You talk a lot about the QC and all the, I think, raw data and this analysis part, but I'm curious how you coordinate, like, for instance, the clustering across multiple labs, or do you have a centralized place, or is it distributed?
So the data is in a central place. When we have very large computation jobs, it's centralized, but it can also happen organically, lab per lab. What I will admit is that the IBL, I thought the experimental part was really collaborative. Right now, the analysis part has not been as collaborative as the acquisition itself. So right now we are still a bit at the beginning in the sense that it was easy for people to work on separate parts of the problems to put this together after the fact. So no big data analysis yet.
All right, let's thank Olivier.